Hello and welcome. This is Dr. Jessie Haft, and I'm super excited to share with you some soil information today to get you a little more comfortable with how soil forms and some of the important physical and chemical and biological characteristics that we look at in soil and utilize for a variety of management purposes. So first, I'd like to introduce you to the factors of soil formation. So these are the agents and forces and conditions that influence, have influenced, or may influence a parent material of soil with the potential to change it. So we have 5 soil forming factors. Hans Jennings created these concepts. We look at climate, organisms, relief, which influences like drainage, material movement through soils, parent material, and time. Excuse me. The main effects of the 5 factors of soil formation are, um, well, there's a lot of them. So let's start with climate and organisms. These are our most active factors. They're the driving forces that cause changes in soil properties. They are, we'll call them the movers and shakers, if you will. And then we have parent material. This is a pretty passive factor that undergoes changes over time. It has differing resistance to change depending on its composition. And a lot of the soil's inherited properties come from the parent material. So when I'm talking about parent material, I'm talking about like the rock that, forms in place to create soil, or the the glacially deposited soil that then over time changes and weathers into what we have today. Topography is another soil formation factor. This is a modifying factor that influences landscape. When we talk about soil climate, there's 2 major parameters. There's precipitation and there's temperature. We usually use the average moisture or temperature values, but sometimes extremes may be more important in soil formation. And really what we're taking a look at here is how much rainfall and how hot or cold it is in a given area. And this is super influential in how soils are formed. We also look at relief and topography, basically where we are at on the landscape. If you look at the diagram here, you can see that across the landscape, we basically call the different portions of the land different names. So we've got, depressions are, these dips in the land where water tends to sit. The upland area is generally upslope, flat areas. We've got the hill slope where we get the actual steeper slopes. We have the terrace that is above usually a stream area. A lot of uplifted terraces kind of form a series of steps that go down into a floodplain and then the stream. Floodplains, let's just take a moment to talk about how we should not build in floodplains because, shocker folks, they flood. The fully developed hill slope is shown here, and this is what we use a lot in soil descriptions for septic purposes. So we've got summits at the top, we've got shoulders where the slope starts to break, we've got the back slope, and we'll have some side slopes sometimes in here. If there's some weird things going on, we start naming this area a little different. It goes down into the foot slope and then the toe slope, and then we break into stream terraces, the floodplain, and then the stream itself. When we're talking about parent materials as soil forming factors, this tends to be a more passive soil forming factor because climate, organisms, those kinds of things are a lot more active and ongoing. However, the original parent material is basically the state of the soil system, the time zero of soil formation. So it may start out as limestone rock, but who knows what it's going to turn into with all these crazy things acting on it. So a lot of inherited properties will be, from the actual forming of the soil. So when we talk about the younger soil, the more evident the properties of the original parent material inherited to the soil. So, for example, when rock initially breaks down into soil, then it's going to be the most representative of what that rock can break down into that it will ever be again. Because once the other factors start acting on it, they change it into the ways that they want. So, for example, organisms start adding organic matter that creates structure, and rain starts leaching all of the minerals and changing the weathering sequences and creating different minerals. And then before you know it, you can't even recognize that it came from shale anymore. Or it may have some properties that were inherited from the shale, but it no longer represents it on a whole. Now it's really more representative of the organic matter and the leachates and the massive amounts of water that have been pumped through it from the climate. Um, in older soils, some of the original features of the initial material are lost depending on the weatherability of the rock. Some rocks are more easily weathered than others, some are more resistant to change. There's different types of parent materials. So the consolidated easy ones to identify, those are like our sedimentary, igneous, and metamorphic rocks. So shale is a sedimentary rock, for example. Diorite is a type of igneous. A lot of people would, call this granite, but it is actually called diorite. And then we have the metamorphic gneiss, which is metamorphized granite or different types of granite. And then we have unconsolidated materials. These are like our coastal plain sediments, our glacial till, our loess, our alluvium, which is water deposited, by the way. Loess is wind-blown silts. And volcanic ash is another example of an unconsolidated parent material that can form soils, but we don't have any volcanoes in Indiana. But I tell you what, if you find one, you let me know because I'd really like to see it. And our fourth soil forming factor is organisms. Organisms in soil are inseparable. Soil serves as the storage bank of nutrients for organisms, and organisms advance soil formation by cycling the nutrients back through it. So there's no significant soil development that occurs without biological activity. So, for example, we've looked at soils in arid soils and really cold regions, and nothing is really happening there. Those soils are like frozen in time. There's not a lot of development that has occurred since the climate became either arid or very frigid. And when we look at other climates, they're primarily the soil changes there are primarily driven by biological activity, and we'll talk about some examples of what I mean here. So here we can see two different environments. We've got a forested environment, lots of trees. We've got grasses in our other environment. And the difference in the soils that we're going to see here are in a tree-based environment, we're going to see a lot more leached horizons. We call these E horizons. Things have been lost from them, washed out, if you will. There's significantly greater amounts of leaching that happens in forested environments, which means we're going to have way less nutrients that are saturated in those horizons. And so we refer to this as having lower base saturation. We also see way deeper cycling of nutrients because of how deep these root systems go. Conversely, when we look at grasses, we're going to see way more organic matter content at the surface from all those fibrous roots just filling those A horizons with just massive amounts of organic carbon. And we're going to have way thicker surface horizons or A horizons because of that. We're also going to see more clay at the surface and way more bioturbation. That's basically the movement or the turning of things in soil by microorganisms or animals and bugs and critters, if you will. These are great examples of some soil profiles from two different types of forested environments. We have coniferous tree environment on the left, and you can see there's a lot more leaching that has occurred in this soil profile. When you look here, you can see this very vibrant E horizon where everything's been washed out of it down here into the lower portions of the profile, where you can see that it's all been just kind of like moved through. And what's left behind is this very light colored, almost ashy looking E horizon. And uh, this ultimately is due to way more leaching from the acidity that comes from pine-based trees, coniferous trees, if you will, the more rainfall that pushes things through here. We're just looking at an exhausted soil profile, if you will. All of its energy, if you will, has been washed out. And so as a mother of twins, I can really identify with that soil profile. And then when we look over here at the deciduous trees profile, our broadleaf, our E horizon over here is way less pronounced. We have a much thicker surface that has a lot more organic matter. We don't see as much leaching here. Okay, nothing has been washed through this profile like it has in the coniferous tree forest. We have much more organic matter near the surface. The E horizon is not as pronounced, and the clay distribution is more even throughout the soil profile. So this is a great example of how just the type of tree present can change soil drastically, just really highlighting the effect that organisms have on how soils form. Most of Indiana, by the way, was formed under either deciduous trees or grassland. So, when we look at further effects of organisms on soil formation, we can look at the effect of microbes. The decomposition of organic matter done by these little boogers is incredible. They are responsible for cycling of nutrients, structural formation. They're the movers and shakers, quite literally, of soil. They also we also see major effects from other animals, like the faunal bioturbation by worms and ants and termites that help to create soil structure. Quite literally, earthworms poop soil glue. And bacteria actually secretes this sugary, sappy substance that glues particles together. And when particles are attracted to the sticky substances on grass roots, for example, they create granular structure. So when we see the disturbance of rodents and other animals as they dig through the soil, this helps break up compaction. They're like nature's tillage, if you will, but in a healthy fashion, of course. We're going to start focusing on some of the things that you have probably seen in soil reports for septic soil testing, and I want to just kind of highlight what some of these things are and what some of the descriptive terms that you may see in these reports stand for, and then we're going to do a little group exercise at the end. So, first, let's start with soil structure. This is just the shape that soil breaks out into, and the more pronounced the soil structure, the stronger it is, the better pathway we have for roots and water and micro and macro organisms to move through the soil pores or little soil highways, if you will, that occur between these PEDs. So the best structure that we can have is granular, and we find this at the surface. It usually forms around roots. The more granular structure we have, usually the more organic matter we have, and so we really like granular structure. It's a great way for water to move through soil quickly and efficiently. Platy structure, not the golden child. Not a way to move water very well. It's like a bunch of stacked dishes, and so water has to meander its way through each plate, moving horizontally just to get a little bit vertically, and then horizontally again to go a little bit further vertically down. And it's problematic, platy structure is. It is our problem child, if you will. And so we don't like seeing it. Oftentimes in Indiana, we're going to see platy structure where we've had tillage occur, and it's usually going to be at the base of the A horizon from the compaction of the weight of the machinery is where we usually find this. And even in no-till situations, or I should say fields that used to be tilled and have undergone no-till, we can still see sections of the plow pan that have platy structure from the compaction. So this is definitely something that we want to remediate when we find it, if possible, so that we can move water through those horizons better. Wedge structure is also a symbol of a problem. We find red wedge structure typically associated with slick and slides. Slick and slides occur where we have a type of clay that has shrink swell properties. Our smectite clays, you may refer to these as montmorillonite. Montmorillonite is just one of the type of smectites. There's several other names, but in this area where we have shrink swell clays, we're going to most likely be seeing montmorillonite type shrink swell clays. And so what happens is when they absorb massive amounts of water, which they're very, very good at doing, if montmorillonite had a superpower, it would be taking on water. But unfortunately, this doesn't result in it being great for leach beds, and I'll show you why. So what we're looking at here is where those clays have expanded so much that they're pressing against the soil horizons around them, and they're basically creating a slipping plane, and that's where this smooth surface is coming in. This smoothed out surface that you see in between each wedge that's created is from so much pressure being applied that it's like squishing parts of the horizon out of the soil profile. And so that slipping plane right there, that smooth surface, that is a slick and slide. And that is what closes soil pores and makes it impossible for water to move through these horizons. When we see slick and slides or wedge structure associated with slick and slides, it is bad news bears for any kind of, leach field. It's definitely something you want to run away from. We also have blocky type soil structures. There's two types of blocky structures. We find these typically in the B horizon or the subsurface of soils. Every now and then you'll find it at the surface of a really eroded soil where it's basically lost most of the surface horizon. If we have subangular blocky, it's got more rounded edges. The higher the clay percentage, the more angular blocky starts forming. That's the one that has sharper edges. And we typically see this in really high clay percentage soils. We can also see prisms form in clay soils where the freeze thaw action or to some degree shrinking and swelling, nothing like what we see with wedge structure. So don't think that when you see prisms that you have shrink swell soils, but it's the slight shrink swell that all clays kind of have a capacity of doing that creates these prisms. It's just a push and pull against the differing portions of soil that kind of create this longer than wide soil structure. And so we see these a lot in the subsurfaces of soils in Indiana. Columnar structure is something that we don't really see in Indiana. It's something that we see in more arid areas, but I just wanted to show it because it's just kind of cool. It forms these like prism-like columns, but the difference from prismatic and columnar is that columnar has a lot of salts associated with these arid portions of the country, and so they form these salt domes, almost like a cap at the top of the shape, and so that's what gives it the columnar shape, is that dome at the top of the column. And so this salt cap is only found in arid climates. So we don't have this here, but I just thought it was kind of fun, so I added it in. We also have structureless conditions. We typically find these in the C horizon. So we have single grain sand is a structureless condition that we can find in like sand dune environments or aeolian environments, which are wind-blown sand created environments. We also have massive, which is a structureless condition. It's basically where the soil just breaks out into giant chunks that are like the size of your hand or larger. It's basically no shape. When you get towards the bottom of a pit and we're pulling these massive chunks of soil out that aren't really breaking down into smaller structural units, that's massive. That's where massive occurs, and that's how you know you're in a C horizon. Or if there's portions of a horizon that are massive, but it's also breaking out in like prismatic or subangular blocky or angular blocky, then you may be in a transitional BC horizon, for example. We also have soil texture. which is the percentage of sand, silt, and clay. This becomes really important for determining loading rates for leach beds, and it's always written in this order: sand percentage first, then the silt percentage, then the clay percentage, and this is what the soil triangle looks like. So we have our percentage of clay on one side, we have our percentage of sand on the other, and then we have our percentage of silt going up the side here. And so it's important to note which side has 0 for each of which way the numbers go for each side of the triangle. So let's practice with loam. We have a 45, 40, 15. So 45% sand. If 0 is over here, we've got 40 here, 50 here. That means 45 is right here in the middle. If you look, these bars are angled this way, so that's the way that we follow this up. Okay, so 45 goes up here. Then let's look at 40% silt. So here's 40% silt and it angles down this way. So it's going to come down through here. Okay, and then 15% clay. So clay has its 0 down here and then it goes up. So here's 10 and here's 20. So we're going to go across here for 15. And where 15 meets up with 40% silt and 45% sand is right here where this orange bubble is in loam. So loam is our texture. The way that we determine texture in the field is by the ribbon method, and I'm going to also show you another method after I show you the ribbon method. So when we make a ribbon, we're measuring the length of it, and typically if it's 1 inch or less, we're down here in this 1/3 of the triangle. So we can be sand, loamy sand, sandy loam, loam, silt loam, or silt. If it's really smooth and buttery, we're over here in the silt options. And if it's really gritty, we're over here in the sand options. If we can't make a shape with it at all, it's sand. If you can kind of start to form a ball, but it doesn't hold together, it's loamy sand. If you kind of throw it up and it doesn't really break apart, then it's sandy loam. And if it has a really good crunch to it and you can make about an inch long ribbon or a 3/4 inch long ribbon, then it's probably loam. Now, once we get over 1 inch, but we're not longer than 1.8 inches, we're either silty clay loam, or sandy clay loam. So sandy clay loam is going to be if we have more sand. Silty clay loam is going to be if we have less sand and more of like a smooth texture to it. And clay loam is going to have more of a crunchy feel to it. When we get above 1.8 inches, that's when we're in our clays. We're in the 38 to 40% clay range and above. So it can be sandy clay, silty clay, or just pure clay. We can also use the wire method to get clay percentages. And with this one, we're going to literally make a soil pencil, if you will. So we're going to roll out our soil so that it's about the thickness of a Sharpie pen or a pencil, and we want it to be about the length of your pointer finger, unless you have really small hands, in which case you need to find a friend with normal hands to, 1, open doors for you, and 2, to figure out how long you need to make your soil wire. Okay, so anyways, with our clay percentage, if we cannot form a wire at all, we're between 0 and 8% clay. If we can form a wire but it can't be picked up without falling apart, we're between 8 and 12% clay. If we can pick it up but it breaks during the process of picking it up, then we're between 12 and 18% clay. If we give it just a gentle shaking, then we're between 18 and 24% clay. If we give it a gentle shaking and the wire bends with some cracks in it, we're between 24 and 30% clay. If the wire only bends with gentle shaking and there are no cracks, we're between 30 and 38% clay. And if you shake that thing and the wire does not bend and it is like solid, then we're at least 38% clay. So I like to use the combination of the ribbon and the wire method to estimate clay percentages in the field, but I would like to note that once you hit 38% clay, there's no accurate method to determine clay percentages above 38%. And so at that point, folks, please require lab tests because you can't accurately predict the percentages of clay above 38%. There's people who can get close, you know, sometimes, um, but it's just one of those things where you're taking a real big gamble if you're not running particle size analysis at that percentage of clay. Soil color is a really important factor that we need to pay attention to for a variety of purposes when trying to prepare a site for, which system is going to be best for it. So when we look at a Munsell color book, we're looking at the hue, the value, and the chroma, and this gives us a lot of information on the soil. So first, the page number is the hue, and we always start in the middle of the book on 10YR. And then when we look at the vertical column, this is the value. And as the value gets higher, the soil gets lighter and grayer in color. I'd like to highlight over here in this column where we have values of 4, 5, 6, 7, and 8. These are our really gray colors, especially in the first two chroma columns. Chroma is our horizontal number. So when our chroma is 1 or 2 and our value is 4, 5, 6, 7, or 8, please note how light in color and how gray this portion of the color sheet is. This is important for redox features, which we are going to be talking about in a few minutes. Typically down here in the 3, 1, and 3, 2, and 2, 1, and 2, 2 colors, this is where we have really high organic matter. Organic matter can mask redox features. We're going to talk more about that later. And then over here in this section is where we have all of our redox oxidation colors. So this indicates some redox features over here where the iron has been oxidized. Oxygen has been reintroduced over here. So we're going to talk more about that in a little while, but first I want to reiterate that we write the hue, the value, and then the chroma. So a 10YR31 color chip is on the 10YR page. We have 3 value and 1 chroma. So this is our chip right here, this nice, beautiful, dark brown color. We always start on the 10YR page, and if the soil is redder than what we find there, we move towards the front of the book where the colors get redder and redder and redder. If the color is more yellow than the 10YR page, we move towards the back of the book where it gets more and more yellow. Rock fragment modifiers become really important as well. When we're talking about soil texture, we are only looking at the percentage of sand, silt, and clay, which sand, for example, is the largest soil texture size, but it maxes out at 2 millimeters. Anything above 2 millimeters is a rock fragment. And so that's what the rock fragment modifiers are. We find in a lot of the glacial soils in Indiana that we have rock fragments that have been jumbled and mixed in and kind of came along for the ride, if you will, when the glaciers kind of scooped out a significant amount of Canada and Lake Erie and dropped it very conveniently here in Indiana. Thanks, Canada. When we're looking at concentrations and other redox features like redox depletions and glade horizons, these are all some terms that you may have heard about soils that become very important for septic systems. So for one, I'd like to start out by pointing out that the gray color in this matrix was not inherited from the parent material. There are some soils that are gray because, they're freshly formed from gray shale, for example. And there may be soils that are bright red because they're formed from ferrihydrite or hematite, which are both very red iron minerals. Goethite is a very orange iron mineral, and so that can give soils those oxidized colors. However, in this instance, what I'm talking about are features that formed from a fluctuating water table. So when we look at this picture, we can see a bunch of gray in the matrix. That's a depleted matrix from water just sitting there for a really long time and reducing the iron there, and the result here is a gray color. And then when the water table falls and oxygen is reintroduced, then this orange color lights up the matrix, and that is an oxidized redox feature. Those are oxidized concentrations from that oxygen being reintroduced. And you can see it again over here too. You can see gray where the water has depleted the matrix, and then we have some redox oxidations here that have orange splotches. And so these are beautiful pieces of evidence from a water table, and we want to pay attention to these so that we know whether or not we need a perimeter drain and where it needs to be placed. So let's look at some more information on this. We're going to do a little activity where we practice finding a water table. This becomes really important. So this is just a typical soil profile. So the depth here is shown in inches. We have an AP horizon that goes to 12 inches. So what that means is that the AP horizon goes from 0 to 12, and then the BTG horizon goes from 12 to 20, and then the BTG2 horizon goes from 20 to 32. So if I were to have done this description, which I did, we would say that one, we have a Glade horizon because of this G, and we have a Glade color here, 10YR 5/1. So where does the BTG1 horizon go from? It goes from 12 to 20. But wait, there's a catch here. Because the organic matter in the A horizon masks redox features, anytime we have a water table that starts at the base of an A horizon, we don't say that the water table starts at 12 inches just because of the redox colors. No, no, friends. We say it goes to the surface because we know that this organic matter is going to mask the water table features that would have risen via capillary action to the surface in this AP horizon. So actually, the water table here actually starts at 0 at the surface. So normally what we look for is concentrations or depletions or a Glade matrix, and that is the horizon that we would start it at. So if we were not at the base of an A horizon, we would say that this water table started at 12 inches because the BTG horizon goes from 12 to 20. But again, since we are at the base of an A horizon, we say it goes all the way to the surface because these dark organic matter features are going to mask the concentrations and depletions that would be occurring in this horizon if it wasn't present. So let's look at another example. What I want you to do is first, I want you to, within the people around you, maybe you're sitting at tables, I want you to tell me the depth range for the BTG1 horizon. What depth does the BTG1 horizon go to from start to finish? I'm going to give you 30 seconds. Do, 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 for those of you Jeopardy fans. Are you ready for me to give you the answer? Okay, so the BTG1 horizon goes from 18 inches to 24 inches. Okay, so the BTG1 horizon goes from 18 inches to 24 inches. And the BTG2 horizon, which I did not ask you about, goes from 24 to 38 inches. So what I want you to do now is I want you to tell me where the water table starts for this soil. We look at the BTG1 horizons. We look at all of our colors, what the matrix color is, the concentrations and depletions, and if they are present. All right, so are you ready? We know that the BTG1 horizon goes from 18 to 24 inches, and that is a glade horizon, and we have redox concentrations. So this is the horizon where the water table starts. However, it is the base of an AP horizon, so we take it to the surface. So the water table here goes to the surface. If we had not had this glade horizon, and we had not had any redox features present in the concentration or depletions column, then the BTG2 horizon would have been the start because it has both concentrations and depletions as well as a depleted matrix. So in that case, the water table would have started at 24 inches because the BTG2 horizon goes from 24 to 38 inches. So hopefully that was helpful for some of you and that you know a little more about soil, and hopefully this will help you understand us weird soil scientists reports as we send them your way. Thank you all so much for listening to me, and I'm so bummed I'm not there in person, but I hope you have a marvelous day. I was just kidding, there's one more slide. You thought you were done with me. Okay, this last slide. I wanted to give you some examples of soil fill. It's basically where soil has been redeposited on a site, and um, we don't want to put systems in soil fill because it's not going to have structure. It's usually going to have human elements. Like for example, here we see pieces of old foundation and bricks all mixed in there. A lot of times we'll find trash, that kind of stuff. So it's not going to move water efficiently. Okay, for real now, I am done and you can end my presentation. Thank you all and have a marvelous day.